Welcome students. Let us study the DNA packaging in the case of eukaryotes. Now in the previous video we have studied that the dimensions of the DNA are such during the resting phase, that means when the cell is not dividing, the DNA is a long thread-like structure which we call it as chromatin, and the length of this chromatin comes out to be approximately 2.2 meters. So if we study the structure of the DNA, we find that the distance between the two base pairs is 0.34 nanometers, and after every 10 base pairs there is a turn. Going by that, we find that the entire length of the DNA fiber, that is the chromatin when the cell is not dividing, it is 2.2 meters, and this is a long thread-like structure. So how this long thread-like structure fits into a tiny nucleus? That is a big question. So the DNA which is present inside the nucleus is a negative charge, is carrying the negative charge. So it is a negatively charged polymer made up of several units of nucleotides. Now this packaging of DNA is very precise, and it is actually done with the help of proteins. These proteins are special proteins which are known as histones. So these histones are proteins which are which are positively charged, and the entire DNA is wrapped around these positively charged histones. Now these histones are positively charged due to the arginine and lysine. This causes the positive charge in the histones, that is the proteins, and around this is wrapped the DNA. All the DNA is wrapped around this ball. Now let us study what are these histones actually. When we study the packing or the structure or how the DNA is being held, we find that there are 5 major types of histones which are associated in the packaging, and these 5 types are H1, we have given different names, H2A, H2B, H3 and H4. These 5 types of the proteins or histones, now we call them, are associated, and out of this the 4 type, that is, I am marking it in underlining with the black, H2A, H2B, H3 and H4, they are arranged specifically to form one round structure which is known as octamer. So the octamer of histones is formed by H2A, H2B, H3 and H4. These octamers are held and DNA is wrapped around this. The blue line over here is the DNA, and these are again held with the help of H1 histone, the marking of red. So if we see over here, this H1 histone is also helping or holding the DNA. Now this is known as linker histone, since it is holding the DNA which is wrapped around this octamer. So if we talk about the structure or the packaging, it is somewhat like a ball, and this ball is known as octamer, which is being formed by 4 different types of histones, that is H2A, H2B, H3 and H4. Around the ball is wrapped the DNA thread, and this DNA thread consists of approximately 146 base pair. This structure, this entire structure which is formed due to this histone molecules and H1 linker is known as nucleosome. It is known as nucleosome, and it gives the appearance of a ball. Now, the nucleosome over here, this is one nucleosome. It is linked to the next one, that is this black line over here, is a DNA strand. And this strand of DNA is approximately 50 base pairs. So one nucleosome and the other nucleosome, they are connected by DNA strand that is 50 base pair, and it is also called as linker DNA. It is also referred to as the linker DNA. Now here the nucleosome consists of 146 or maybe around 200 base pairs, and the linker consists of 50 base pairs. So, as the DNA which is being present in the cell when the cell is not dividing, due to the presence of the nucleosome and the linker DNA or the DNA strand, it gives a beads on string like appearance. It is somewhat resembling the beads, like suppose this is the DNA strand, these are the nucleosomes. So they appear like beads on the string, and the string is actually between the two nucleosomes is the linker DNA, and that is 50 base pairs, consisting of 50 base pairs. So this is a beads on string structure of the DNA which is not dividing. Now the H1 histone performs certain important functions. That is, it helps in the chromatin compaction. When the cell is not dividing, the chromatin fibers are present within the nucleus, and these chromatin fibers are in the form of beads on the string because of the nucleus. Because of the nucleosome. So the H1 histone actually holds all the DNA string which is present around the nucleosome. It is during the division, it helps in the chromosomal spacing, also in the nucleosome spacing. So we find that in the case of eukaryotes, the DNA is present, or the model, the structure which it presents is a bead on string model or the structure, and the octamers. Octamer is formed by mainly the 4 type of histone proteins, that is H2A, H2B, H3 and H4, and H1 protein holds the octamer together. So this octamer of 14 histones appear like a ball. When the cell divides, now when the cell division starts, these nucleosomes come closer to each other and they form compact structure. During the metaphase and the other phases, the compaction of the chromosome take place, that means due to the supercoiling, and then the cell is ready to divide.